Good afternoon, everybody, and welcome to this Darwin Tree of Life session in Biodiversity Genomics 2020. My name's Alex Twyford from the University of Edinburgh, and I'll be chairing this first session on strategy and sampling. And then in an hour's time, I'll be handing over to Peter Holland from the University of Oxford, who will be talking about sequencing, assembly, and data. The Darwin Tree of Life project is this visionary project to sequence reference genomes from all species in Britain and Ireland. And it's a Wellcome Trust-led initiative that involves very many partners, and we'll be hearing from lots of different partners throughout the afternoon. I'm excited to start by introducing Professor Mark Blackster from the Wellcome Sanger Institute. He's Programme Lead for Tree of Life and Senior Group Leader at the Wellcome Sanger, and he'll be giving the first talk on the Darwin Tree of Life project.
Hi there, I'm Mark Blackster. I'm lead on the Darwin Tree of Life project. I'm based at the Wellcome Sanger Institute, and I'm here to present this introductory talk in the session on the Darwin Tree of Life project to explain why we're sequencing all the eukaryotic species that live in Britain and Ireland. And we called it the Darwin Tree of Life project, taking inspiration from Darwin. Darwin spent much of the latter part of his life down in Kent and described the amazing diversity of life on this planet and the processes, some of the processes that generate it, by working in his back garden. And that's what we intend to do for this project here, by working in our back garden to actually start on the great project that is the Earth Biogenome Project. You may ask why work on this small archipelago off the west coast of Europe, which we know as Britain and Ireland, because we're not exactly known for our hyperdiverse ecosystems. We're not known for having lots of large, exciting wild animals. But there are several things about our islands which recommend them to us as a starting place, a perfect laboratory, if you like, for doing a total biota genome project. The first is that only 14,000 years ago, almost the entirety of the islands were covered by ice during the ice ages. But by 12,000 years ago, an ecology had restored itself, had reassembled itself over much of the islands, such that the ecosystems we know were reconstructed. So this is a great model for how ecosystems can evolve and change under climate change, obviously very relevant to today. Another major effect on the biodiversity of our islands is the impact of humans. So anthropogenic change has really changed the ecosystem that all the organisms on the islands live in. And especially we're really interested in the effect of this in terms of agroecosystems on organismal evolution, but also the role that invasion from other countries and other ecosystems can play on defining an ecosystem. A third reason for working on this group of islands is because we know so much about the biodiversity. This here is a map of the locations of golden eagles in Britain. And we have this for most of the species in the British list. So we know where they are. We have historical records of their diversity and their numbers. And so it's exquisitely understood where and when species are. The other thing about the British biodiversity is that it's known in great detail. So we have probably the best described species list of all ecosystems. There are only about 62,000 species. But excitingly, these species correspond to a large component of the phylogenetic diversity of life on the planet. So 62,000 species is only 4% of all the species described in the world, but they correspond to about 40% of all the families. So by sequencing Britain and Ireland, we should be able to cover about 40% of the phylogenetic diversity of life on this planet. So I work at the Sanger Institute, which is well known as a sequencing institute for sequencing human DNA and others. But we're not a biodiversity institute. We don't have a record in biodiversity. And so we built a collaboration, which we call the Darwin Tree of Life project, which is now funded by the Wellcome Trust, to do this sequencing of everything in Britain and Ireland. This project includes biodiversity partners like the Natural History Museum, Royal Botanic Gardens in Kew in Edinburgh, Marine Biological Association and the University of Oxford's White and Woods field site, and analysis and sequencing partners. So University of Edinburgh, University of Cambridge, Earlham Institute, and EMBL EBI as well as the Sanger Institute. So our project started just less than a year ago with a discretionary award from the Wellcome Trust and with in-kind contributions from all the partners. So what we're going to do? Well, first of all, we're going to try and sequence a reference quality genome for every family of organism that's represented in Britain and Ireland. That we think will take us about 3 years. Then we're going to move on to do a reference genome for every genus and then for every species. So we hope this will take us about 12 years. To do this, we really need to work together. So we need taxonomists to, natural historians to collect and taxonomists to identify all the species. We're going to assist that and build on that using DNA barcoding. We then need to extract DNA from every single species, no matter how big or how small or how difficult they are, and generate really high quality data, which can then be assembled using the best quality tools to give really high quality assemblies. And we're then going to release those publicly. So in terms of data generation, we really want to get to chromosomally complete assemblies. And so we need really high quality data and also very long linkage between sequences. And so we're going to use long read data at the moment, largely from Pacific Biosciences Sequel II Hi-Fi, and long range data at the moment, largely from Hi-C data. We're also generating RNA-seq data to annotate each one of these genomes to identify the genes. So I've mentioned the desire to do chromosomally complete genomes. This is a representation of the genome of the ringlet butterfly, Aphantopas, and it demonstrates what we can do with chromosomally complete genomes that we couldn't do otherwise. So this butterfly has, compared to Bombyx mori, the silkworm, the silk moth, 2 chromosomal fusions, which we'd only find out, we can only identify now that we've got a chromosomal assembly. And it's this sort of new biology we want to get at with these chromosomal assemblies. Historically, it's often taken a while for genomes to get into the public domain, but we want to change that as well. And we want to publish every genome as soon as we generate it, before we do analysis. So we're going to release them all as publicly accessible genome notes. These genome notes are a bit like the ones that are already normal in the bacterial genomics community, and will be openly accessible as soon as the genome is generated and deposited in the public databases. And these will announce to the community that the genome is available for them to use for whatever, whether it's conservation genetics, whether it's investigating new biology, or whether they want to mine these genomes for biotechnological purposes. So you're going to hear some more talks in the following sessions from people in the Darwin Tree of Life project about the processes we've put up and some of the first data from the project. This project is really based on a large number of people putting a lot of effort in. This is a current list of people who've collected, identified, extracted, sequenced, and assembled the data. The list is growing all the time. So sit back, listen to the talks, and I hope you enjoy this Darwin Tree of Life session.
Thank you very much, Mark, for a really exciting talk. And if anyone has any questions, please do post them in the chat. While those questions are coming through, perhaps I could kick off, Mark, by asking you, we heard from Harris Lewin yesterday in his plenary talk about how people are looking at Darwin Tree of Life and some other projects to learn some lessons about how to perform this type of research at such scale. Maybe you could start by telling us a little bit about what you've learned about getting sample acquisition at scale from Darwin Tree of Life so far in the first year of the project.
Well, obviously, this first year has been a very unusual year. It's been a plague year for the world. So we have been collecting in our own back gardens. What's really amazed me about this project so far, and especially with our biodiversity partners, is the enthusiasm with which all the partners have embraced the idea of doing genomics. I think traditionally, there's been some skepticism of genomics because we largely have worked on model organisms, or in a way the genomicists want to come in and extract the DNA and go off and do some interesting stuff of their own. But really we've had huge buy-in from biodiversity partners and people working with these organisms in the wild, such that they see the value of these genomes coming back to them. So people have been able to collect despite coronavirus, and people have been delivering samples to us. We're going to hear from the samples collection folks soon, but we have about 700 species frozen and in the Sanger Institute ready to sequence already. And we know, because we talk to our partners frequently, that there are another 700 or so collected and sitting in their freezers waiting to ship to us. So we've started, we're only a year in, we're slightly behind because of coronavirus lockdown, but things are progressing really fast.
Great, thank you. So the questions are now starting to come in thick and fast, and the most upvoted question so far is, I'd like to know how the team will deal with endemic and threatened species, if it applies, of course, and that's an anonymous question.
Yeah, so one of the things we have been very, very keen to make sure we're doing is that we're doing everything ethically and responsibly. And we have developed a comprehensive ethical code of conduct in terms of sampling. And this means that everything we collect has to be permitted. So there are all sorts of levels of permits required, from global legal ones to do with truly endangered species, of which there are a few in Britain, through locally endangered species which have what we call local biodiversity action plans for them, but also things like making sure that if we sample from an area that we have permission to do so from the landowner. And so that set of permissions, I think, means that we're going to be sampling ethically as possible across the biota of Britain. We really want to sequence the endangered and the threatened species, because often the genomes of those will be very useful going forward for the people doing conservation biology on those. And so we are actively looking to sample ethically as far as possible. So it's all being very carefully organized, and we do not want to damage or threaten any species we work on.
Great, thank you very much, sir. And I'll ask one last question before we move on to our next speaker. And that's now our top voted question now, is by Rob Waterhouse. RNA-seq to support annotation, what strategy to get multiple tissues and or life stages, or are you just going to take what you can get? Long read or short read RNA-seq? Okay,
so two questions in there. The first one about the tissues we get, obviously from some organisms, microdissection of tissues is not going to be possible, and so we're not going to do that. So it will be total organismal RNA-seq from, for example, a small beetle. And for the organisms where we can get separate tissues, we will. But in general, we are only using these data to gene find and not to do any functional genomics. So we will go for tissues that we believe have high transcript diversity, brain, testis, liver, for example, from vertebrates. In terms of the technology we use, there's a R&D process in place looking at the relative merits of long-read RNA-seq versus short-read RNA-seq. The short-read RNA-seq at the moment giving us much greater density of sampling, so actually allowing us to identify many more rare transcripts, and the long-read RNA-seq giving us the ability to string exons together on the genome. So there's a tension between those two, and we're doing the research to try and work out which is best now.
Great, thank you very much. So it'll be really exciting to see a lot of that transcriptome data coming through. Really interested to see those gene annotations. So really exciting. Thanks again for a great talk. So we move now to our second invited speaker of the session. This is Ilya Leach, who is Assistant Head of Comparative Plant and Fungal Biology Department at Royal Botanic Garden, Kew. And she'll be giving a talk entitled Sampling Plant Genomes for the Darwin Tree of Life Project and Why Size Matters.
Hello, everybody. Thank you for logging into the session, and thank you also to Alex and Peter for their kind invitation to speak at this session. And as you can see from the title of my talk, what I'd like to do is to give you a brief overview of our progress and strategy for sampling plant genomes for the Darwin Tree of Life project, as well as to explain to you why genome size matters, why we're collecting genome size data for every species that we are analysing. Just as a brief bit of background, as many of you may be aware, the British flora is perhaps one of the best studied floras on this planet. And this is largely a result of having benefited from many centuries of of documenting and studying the diversity, distribution of species, their ecology and evolution. And I guess the real overall aim of this part of the Darwin Tree of Life project is to bring together all this wealth of information that we have on the British flora, and to combine this with the developments in DNA sequencing and analytical approaches being developed by the Darwin Tree of Life in order to enable us to produce and establish the world's first genome sequence flora. So that's our overall aim. In terms of species numbers, the British flora is not particularly species rich. It comprises around 2,600 native species, distributed around 251 families. Although you can see it includes representatives from all the major lineages of land plants, as you can see here in this slide. And then as far as our targets for this year, we are aiming to collect 235 species from within the British flora. And in order to identify, well, these ones are going to be sent off to Sanger, and in order to determine which species that we want to collect, we have been working with the plant working group in order to prioritize them. And we've identified 3 priorities that we want to target. So the first is to maximize phylogenetic diversity, so collect species from as many different families as possible. Secondly, to target species with small genomes and that are diploid, where we have that information available, because we think this will considerably simplify things for the Darwin Tree of Life as they work towards optimizing their pipeline for generating these high quality chromosome level genome assemblies. And then finally, our list predominantly is consisting of native species, although it does also include a few non-native, such as those of medicinal interest or plants which are particularly invasive for the British flora. So in terms of numbers, what this means is that we are aiming to collect 139 vascular plant species and 96 species of the bryophytes. And so how are we progressing? Well, as you might imagine, in these sort of COVID-filled times, actually, for a lot of the year, we were not able to collect anything, but we were very fortunate with the easing of lockdown restrictions in the summer that we could employ the expertise, taxonomic expertise and fieldwork experience of 2 botanists, Martin Christenhuth, who was based at Kew, and Zoe Goodwin, who was based in Edinburgh. And they were tasked with going out to select suitable plants within and around our 2 botanic gardens. And you can see on the right here just some examples of some of the plants that they did manage to collect. And then for the non-vascular plants, the bryophytes, we have been fortunate again to have the expertise of David Bell and David Long, based in Edinburgh, who have also been out collecting some of those bryophyte lineages, the mosses, liverworts and hornworts. And you can again see some examples of some of the beautiful mosses that they have been able to collect so far. So in terms of numbers, we have so far managed to collect 190 out of those 235 target species. And there's good phylogenetic diversity distributed across 117 families. And what you can see from this, on the right-hand side of this slide, that already we've exceeded the targets actually for the vascular plants, and for the non-vascular plants, we're also making good progress on, and this is likely to improve, probably reaching our targets by the end of the year through ongoing field work, and as well as taking advantage of the excellent bryophyte collections growing in the Royal Botanic Garden in Edinburgh. And then as for what we're collecting for each species, then we are collecting multiple samples to give Sanger plenty of material to work with in order for optimizing the extraction of high molecular weight DNA that they need for their high quality genome assemblies. But we've also been collecting herbarium vouchers, silica samples for DNA barcoding, associated metadata such as photos and GPS coordinates, as well as fresh leaf samples for genome size. And so this sort of raises the question, why do we need this genome size data? Well, the answer to this question probably lies in the fact that, well, we know that across eukaryotes as a whole, genome sizes have been shown to range 64,000 fold. So there's huge diversity across eukaryotes. But actually, most lineages of eukaryotes are characterized by narrow ranges of small or very small genomes. And you can see just 3 examples here where we've got reptiles, birds and mammals, narrow ranges of small genomes. Well, the situation in plants is completely different, because here within plants, particularly the flowering plants, what we see is a huge diversity of genome sizes, probably the biggest range for any comparable group of eukaryotes, with genomes ranging 2,400 fold in size, from the smallest genomes found in some tiny little carnivorous plants belonging to the genus Genlisea, such as Genlisea aurea shown here, whereas the biggest genomes so far reported not only for any plant, but actually for any eukaryote found in this plant here, Paris japonica. And you can see its genome is coming in at 149 gigabases of DNA. So what this means is actually, if you remember that our own genome is just 3 gigabases, Paris japonica actually contains 50 times as much DNA in each cell compared with our own genome. Well, that's the diversity across plants as a whole, but even within the British flora, based on our currently available data set, we can see that genome sizes also vary considerably, around 1,200-fold, from around the smallest so far being found in this lycophyte, Selaginella selaginoides, and the biggest genome found in mistletoe, Viscum album. And you can see its genome is certainly a very big genome as well, having around 30 times as much DNA in each of its cells compared to our genome. Well, this genome size diversity certainly has practical implications for projects such as the Darwin Tree of Life, because what we know is that if your project is aiming to assemble whole genomes, you really need to know the genome size in order to be able to determine how much DNA sequence data do you need to generate in order to get sufficient coverage and hence to ensure robust genome assemblies. You can imagine it'd be a bit of a shock if you were to start sequencing the genome of Viscum album if you had no idea just quite how big its genome would be. But this is one practical implication, but I also just want to highlight another aspect of plants, and that is the occurrence of intraspecific ploidy diversity. So that is the presence of different individuals of the same species having different ploidy levels. And here is an example in a grass, Agrostis solanifera, where 8 different ploidy levels have been reported within this one species. Now, you can see it's very easy actually to use genome size estimations to distinguish between these different ploidy levels, because something like a tetraploid will have double the genome size of a diploid. And so we are also measuring genome sizes because we want to be able to distinguish between these ploidy levels in species which show this diversity, and hence ensure that wherever possible, that the individual plant that is selected for sequencing is a diploid, because that will considerably simplify the sequencing and assembly for that particular species. And in fact, I hope then you can see that there's a good reason for why we need to measure the genome sizes for all the samples that we're collecting. And we're particularly fortunate to have the skills of Sahar, based at Kew, who will be doing this work using the technique of flow cytometry to measure the genome sizes. Well, so there are the practical implications of genome size diversity, but what about the biological implications? Does it matter to a plant whether it's got 2,400 times as much DNA in each of its cells compared to its nearest neighbor? What impact does it have on the plant? Well, the answer is yes, it does have an impact. And for the last few minutes, then I would just like to give you a brief overview of these biological implications of genome size diversity. So certainly, it's been long known that genome size has an impact at the nuclear and cell level. So for example, if you look at this top graph here, what you see is that the positive relationship between genome size and nuclear volume means that the bigger the genome, the bigger the size of the nucleus. Well, that's not surprising. You need a bigger nucleus to contain all that extra DNA. We also see that there's a positive relationship between genome size and, for example, the duration of meiosis. So the genome, the longer it takes for plants to go through cell division and to grow and divide their cells. And then at the cell level, we also see positive relationship between, for example, genome size and guard cell length. The bigger the genome, the bigger the guard cells. Well, these relationships that we see at the nuclear cellular level are known to have a knock-on impact at the whole plant level, influencing where and when and how, for example, a plant is able to grow and compete in the environment. And this is a simple, quick example of how this impact at the nuclear level, cell level, can impact the whole plant level. I just want to show you this this relationship of genome size and guard cell length to show you how it can impact on a plant's ability to grow and compete in water-stressed environments. And this is because what we do know is that species with small guard cells show a much more rapid response to water stress than those with big genomes. And so perhaps it's not surprising that in water-stressed environments, such as desert-like environments with low rainfall and those in epiphytic plants, for example, that here the species that are able to grow are characterized by small guard cells. But because of that relationship between genome size and guard cells, we see that also the plants growing in these environments also have small genomes. whereas species with big genomes appear to be excluded from these sorts of environments. And indeed, these, as the number of studies have increased, it is clear that actually having a big genome appears to impose particular constraints on where plants are able to grow and compete. So I've just shown you, for example, how having a bigger genome can restrict your ecological options in terms of the ability to compete in water-stressed environments, but other studies also suggest that in soils with low levels of nitrogen and phosphorus, here also species with big genomes appear to be excluded. They're not able to compete successfully. And then other studies suggest that species with big genomes are restricted to being obligate perennials. They simply, because of that relationship between genome size and cell cycle time, they're simply not able to grow quick enough to complete all their cell divisions within a year to be an annual, and they are restricted to being obligate perennials. We also see that plants with big genomes are restricted in their life strategy options. There are no big genomed weeds, for example, and they are more susceptible to pollution and radiation, and in some cases, reduced speciation has been observed in some lineages as well. So really taken everything together, what we see is that genome size is certainly having an impact, and therefore that genome size matters. It's impacting the ability of plants, for example, maybe to how they're going to respond to environmental changes, as well as their evolutionary trajectory. So I hope I've convinced you that genome size indeed is important, not just for the practical implications, but also the biological implications that I've just been showing you. And the new genomic data arising from the Darwin Tree of Life project is going to really be invaluable for providing us with deep genomic insights into the underlying genomic processes contributing to the diversity of genomes that we see in plants. So it remains only for me just to thank everybody, particularly those at Kew in Edinburgh who have helped collect all the plants that we've managed to do so far, as well as everybody on the Darwin Tree of Life Plant Working Group. So thank you very much.
Thank you very much, Ilya, for a very stimulating talk. And as we're waiting for questions to come through, please do type your questions. I'll kick off by asking, with great variation in genome size in flowering plants also comes great variation in repeat content. I wondered what you felt we could learn from studying that diversity of repeat content across the British and Irish flora.
Well, I think, as you say, certainly the bigger genomes, the more repeat content there will be. And I guess what we're interested in some of the research we're currently doing is exploring actually how the dynamics of repeat evolution might vary across genomes of different sizes. And we're finding indeed that for some species with big genomes, their repeats are evolving in a quite different way than they are for species with small genomes. But that's based on just a few species. And I guess by having this diversity, as I mentioned in my talk, we already know there's a 1,200-fold variation in genome size across the British flora. We shall have much greater insights to be able to explore this in a greater detail about what it actually means in terms of how does the genome size itself impact on how the genome is evolving, particularly the repetitive fraction of it.
Great.
Does that help?
Yeah, thank you very much. So we've got lots of questions coming in, lots of engagement in the chat. So our top voted question at the moment is by Thomas Keane, who is asking whether genome size is mostly driven by transposon activity and copy.
Certainly in terms of, I was just mentioning, of course, that we've got these different dynamics in how the repeats are evolving in genomes of different sizes. What we do see is that in species with small genomes, that their repeats are certainly being driven, that repeats are certainly contributing a lot to the evolution of genome size and giving rise to larger genomes by the activity of those repeats. Whereas what we see in the genomes which are much larger, above a genome size of around 10 gigabases, we see that the repeats are turning over much more slowly. They don't appear to be eliminated from the genome as efficiently as they are in species with small genomes. And here the genomes are big, not because of the activity of the repeats, but because they are just slowly degrading, because they're not being eliminated from the genome, they're slowly degrading. So these big genomes are really just a mass of sort of degraded, dead and decaying repeats scattered across the genome. So those are differences in what's giving rise to the big genomes.
Yeah, that's great. And I'll ask you one final question before we move on to our next speaker. And we're getting similar questions from a few people here. Rob Waterhouse and Laura Kelly are both asking similar questions, which are about sequencing the very large genomes. And are we, for Darwin Tree of Life, are you holding back on sequencing the big genomes, or are you targeting them because they're a particular interest?
From my own personal perspective, I'd like to be targeting them completely, because I think that's where we'll find some really interesting things going on. I mean, up until now, the largest genome so far sequenced with a whole genome sequence is only around 31 gigabases. Whereas if I already mentioned that the mistletoe is coming in at over 90 gigabases. So I think there'll be lots of exciting things there to learn from that. And so that's my perspective. I think from Sanger's perspective, they might feel rather differently, certainly at the beginning, because they are probably going to be very challenging with those vast amounts of repetitive DNA sequences in the genome. So I guess it depends on who you ask as to how successful we are to get as many big genomes in there as possible.
The big genomes certainly get my vote. That's all I can say.
One down.
So we'll thank you very much, Ilya. That's really, really interesting. So we'll now turn to 3 short talks. And for these short talks, I'd really encourage you to get your question in during the talk so I can ask your question as soon as possible. So the first of these 3 short talks is by Lyndall Pereira, who is sample coordinator for Darwin Tree of Life project at the Natural History Museum. And she'll be giving a talk entitled How to Sample the UK Fauna and Flora for Darwin Tree of Life.
Hi everyone, I'm Lyndall from the Natural History Museum, and I will be chatting to you about how to sample the UK fauna and flora for the Darwin Tree of Life. So the project involves a network of 10 key partner institutions with the aim to generate data that is not only relevant, but revelatory and can be used immediately. Species are prioritized and must have clear value to scientists, to the public, and to agencies with responsibility for biodiversity management. So in the first phase, the goal is to generate complete genome assemblies for reference species for over 3,000 families. And in the next 2 phases, it's to generate this data for all eukaryote species of the British Isles. So this data can then be used to understand in-depth British biotic diversity in numerous contexts that are and will be relevant to our society now and in the years to come. So for the first phase, the aim is to collect 8,000 species, 4,000 of which will be genome sequenced by 2022. And so that means we have 60,000 species to choose from. So how do we decide in a meaningful way? So of the 60,000 eukaryote species, we look at the first priority, which is phylogenetic coverage, and that takes us to over 4,000 families. And this represents 1/3 of families on Earth, which is a good basis for global projects. So now we need to exclude those that have been sequenced already, and at least for the first phase, those that are very small, hard to find or capture, or extremely rare or thought to be extinct. So this takes us to about 3,000 families. And if we add other priorities that include research interests, iconic species, important species, protected and endemic species, as well as targetable species, what is actually feasible to collect and find? And this takes us to a total of 4,000 species for the first phase. When it comes to sampling, it requires an intense amount of collaboration and coordination effort. Species lists not only need to be updated and maintained, as they are constantly changing, but they need to be transformed into a target list that is within the scope of this project. Then the species need to be found, and in some cases identified live by experts. So we need to find and engage with these taxonomic experts. Specimens need to be processed correctly, and extensive metadata is generated during this process and needs to be trackable from the field to the lab. When it comes to processing, every group presents its own challenges, and in some cases, dissections need to be done in field and on dry ice to maintain the quality of the tissue. DNA barcoding is also essential in this process and before any genome sequencing can take place, as it not only affirms the taxon, but it also provides a tracking identifier throughout the sequencing process. And then an absolutely vital part of this project and this process is verifiable vouchers, which come in the form of DNA extract, tissues, photos, and in some cases, physical diagnostic organism parts. And these are stored and curated in permanent collections for the long term and can be used to validate the research we do now in the years to come. So by the end of this pilot phase, we'll not only have high quality genomes for 4,000 key species, but we would have developed the tools for completing the whole of the Darwin Tree of Life. And by continuing to expand our collaborations, intensifying effective and efficient sampling, the project will be able to train and inspire research scientists, conservationists, and natural historians for generations to come to make effective and transformative use of the data in ways that we haven't even considered yet. And with that, I would like to thank everybody who's involved in this amazing project. Thank you.
Thank you very much, Lyndall, for a really enjoyable talk, and great to see recognition of amateur naturalist societies as part of your talk, which is a really exciting part. While any questions come through, I wonder if I could ask you whether you have a feel for how many animals might be too small to sequence with current sequencing technologies.
Sure, how many? Well, I guess it's anything that's smaller than about 5 millimeters. So for a lot of the arthropods, that's, I don't know, I'd imagine about at least a fifth of the families that are there. But I can't be certain yet. But I guess we'll be releasing this sort of information quite soon when we have our priority lists fully populated.
Great, and actually that leads on perfectly to the top voted question at the moment. So I think I'll take this one question, and then if time allows at the end of the session, I'll take some more questions. So the top voted question is there a list of species and their priority somewhere public or shareable? So can we see if our samples are useful to provide?
Yes, so this is something that we are working on, and there should be a portal, at least, that Sanger will provide, and also from the NHM on our portal as well. So when we have this list ready to go, it will be available for everyone to see. There is a survey that can be filled in for research interest, which I should have put in my talk, but now I think I'll put it in the Slack space. So if anyone is interested, I'll put all of that information that we do have up there, so we know what everybody is interested in. But yes, so soon that priority list will be out. It does change and update depending on research interest. And yeah, so soon. It'll happen.
That sounds like a really useful resource, and we can also retweet that on the Darwin Tree of Life Twitter feed so we can get some wider engagement there. Yes, so thank you very much, Lyndall. And sure. We'll now turn to Liam's talk. So our next speaker is Liam Crowley, who's a postdoctoral research assistant at the Department of Zoology at the University of Oxford. And Liam will be talking about genome sequencing and ecosystem, the arthropods of Wytham Woods.
Hello, I'm Liam Crowley, and I'm a postdoctoral researcher at the University of Oxford, and I'm working on the Darwin Tree of Life project. Thank you for inviting me to speak today. Specifically, at Oxford, we are working on the Wytham Genome Project, whereby we are, as part of the Tree of Life project, collecting, processing, and identifying arthropods from Wytham Woods. We are interested in, as part of the wider project, gaining 1 or 2 species from every taxonomic family of arthropods for full genome sequencing. But the unique thing about Wytham Woods is that it's the site of some of the longest running ecological experiments in the world. So we are able to put these species within this greater understanding of the ecological context and identify species and groups of species of particular evolutionary and ecological interest. So to select species, we assess them against a series of selection criteria. And these are both around the selection for the taxonomic representation, but also for our deep dives of species of evolutionary and economic interest. So this is based against a series of criteria such as, first and foremost, the community survey. So we asked the research community in the UK which species they would most like to see full genomes for. And of those, we have already collected around 20% of the species suggested. And there are several other concerns based around the practical and logistical constraints, such as how easy and realistic is it to collect and identify these species, and other practical concerns such as if all being equal, we'll go for species with perhaps a larger body size, or whether there's a transcriptome already available. So in the initial phase, looking for low-hanging fruit. Of the species that are identified, of particular groups or species groups for our deep dives, these are largely based around either a taxonomic group, so for example, the moths and the hoverflies, or around an ecological community such as the dung fauna or the oak 100, which are 100 species, key species associated with oak trees. So these are just some of the deep dives which we've started so far. Day-to-day work on the Whiteham Genome Project largely focuses around the collection, identification and preservation of arthropod specimens to go to Sanger for the full genome sequencing. Collection is based within the 1,000 acre wider Whiteham area, and this covers actually a range of habitats, not just woodland, lots of different type forest stands of different age and management histories, but also areas of calcareous grassland, fens, wetlands, and even agricultural areas from the university farm. The identification has to be around live specimens to go into the processing step, so we can snap, freeze and preserve high quality DNA. Therefore, this presents a lot of challenges, particularly for difficult taxa. So initially, we started off with the low-hanging fruit species, which we could confidently identify in the field. However, we're also investigating other techniques such as knockdown techniques with cooling or CO2 to identify under the microscope live specimens. And even excitingly, with some specimens, we were able to identify based off key morphology, so able to dissect off body parts such as genitalia to be identified in traditional ways during the preservation step. In the preservation, we are using dry ice to rapidly cool specimens and putting samples into uniquely barcoded fluid X tubes. So the specimen can go into the cooling arena here, and then we can quickly photograph and dissect out the relevant parts. We're taking one or possibly more leg for smaller specimens to go for barcoding as a confirmatory step. And then the specimen is then broken down into different tissue parts, which can go into different techniques, such as PacBio and Hi-C. Our progress, we have so far collected, identified and preserved 692 species of 158 families across 17 orders. You can see some of the key orders here. Notably, the orders associated with these deep dives have the best coverage, such as the hoverflies for the Diptera and the moths for the Hymenoptera. And then we have assessed the 925 UK families of arthropods, and we have already collected 158 or species representing 158 of those families, and identified a further 320, which we would optimistically expect to be able to find at Whiteham, with a further 228, which we be unlikely to pick up at Whiteham. And then finally 219, which we have not assessed, as these are the Acaro, the mites. So with that, I'd like to thank you for your attention and invite any questions.
Thank you, Liam, for a really interesting talk. And while the questions come in, I'll perhaps ask you, you mentioned at the start of your talk that you've got lots of ecological data as well for the woods. I wonder whether you could tell us a little bit about that and how you might be able to merge that with some of the genome data that's coming through.
Yeah, so as I said, this is a remarkably well-studied woodland, and there's research projects going back pretty much over the entire 80-year history since the woods have been owned by the University of Oxford, in all different groups, from badgers and small mammals through to long-term ecological plots monitoring plant species. So we're trying to identify communities of particular interest that we can then try and marry up some of the genomic data with some of the ecological interactions You'll be able to hear a little bit more about some preliminary stuff in the next session, DToL 2, with some of the other work from Oxford.
Great, looking forward to that. So we're getting some questions through. One of them, and I'll just ask one in the interest of time, is have you found any new species records for these words, I suppose, in the process of doing your sampling?
Yes, actually, we've had a couple of really exciting records turn up, and we have been able to collect and process these species for the project. So I think by having such a remarkable site to base the project in, it's a real double win in terms of generating specimens of species which otherwise would be very difficult for us to come across and could potentially have some really interesting implications further down the line.
Great. We're getting lots of other questions through, but I'm afraid we've got to move on. But yeah, our next and final speaker before we take a break is Caroline Howard. Caroline is Scientific Manager for Tree of Life Genomics at Wellcome, and she'll be giving a talk entitled Wet Lab Workflows to Generate High Quality Genomes for Darwin Tree of Life Project, Present and Future.
Hello, and thank you very much for the opportunity to present at the Biodiversity Genomics Conference 2020. My name is Caroline Howard, and I'm the Scientific Manager for the Tree of Life programme at the Wellcome Sanger Institute. And I'm going to give you a very brief overview of the wet lab workflows that we use to generate high quality DNA and RNA for genome assembly. So the Tree of Life at Sanger currently comprises of 3 major projects. There's the Darwin Tree of Life project, the Moore Foundation Aquatic Symbiosis Genomics project, and the Vertebrate Genomes project. And we fully anticipate that more projects will come under this programme in the future. The requirements and the goals of all of these projects mean that we need to be able to extract high quality and high molecular weight DNA from a broad range of taxa and also RNA. So 2 work streams immediately become apparent. First of all, we need to be able to have a high throughput of relatively simple samples, and we also need a real R&D focus on the more difficult and more challenging organisms. So here is an overview of the routine throughput in our DToL lab, Tree of Life lab. The samples come into the laboratory from here. That's thanks to the work of our amazing samples management team and all of the collaborating institutes that are in many different countries. Once the samples arrive with us, they go through the cryoprep disruption process. We then have a standardized high molecular weight DNA extraction protocol that we used that's adapted from a MAGATRACT method. We then do the quality control of the extracted DNA, and we use the qubit to do this, also the nanodrop and the femtopulse. So here we need to get a really good picture of both the quality and the quantity of DNA that's been extracted. After this, we can then prioritise the library prep based on the yield and of course the quality of those extractions, and quite often we do find that repeats are necessary. So we follow back to this stage of the process. Once we have the sufficient yield and quality of DNA, we will go through to the library prep processes. And here is where we hand over to some of our very specialised teams within the Sanger Institute who deal with the particular sequencing platforms and technologies and are critical in collaborating with us to develop these protocols. So we currently deal with ONT sequencing, 10x library prep methods and PacBio library methods as routine. And we would be looking to do each of those in parallel. Also, we need to do a high C library prep, and that needs to come from the raw tissue from the samples in here. We can't do the cryo prep currently before doing the high C library prep for these materials. We would also like to be using the cryo prep material to do RNA extraction and the subsequent quality control of the RNA through to sequencing. And then all of these methods come together to be combined in our assembly and curation methods. So the routine throughput that I've just described is a fairly simple and fairly nice process flow. However, we need to do this from one individual from each species. That requires an extremely good quality extraction with high molecular weight DNA featuring as the main component. Currently, we require a yield of approximately 1,500 nanograms of high molecular weight DNA. We will be looking in future to reduce that number, but at the moment, that's where we are. We also need to have some tissue sample remaining for the high C preparations, and again, some tissue remaining for the RNA isolation. So as you can imagine, when the individual is extremely large, this isn't an issue for us, but as the size of the individual or the organism reduces, this can become quite difficult for us. Here I've just given an example of the type of quality DNA that we would be looking for. So this is a femto profile from one of our extractions. You can see that there's very little low molecular weight DNA here at the bottom. And we have a very clean, sharp peak here, which is approximately 190 kilobases. That's the type of quality that we'd like to see for all of our samples going through to all of the pipelines. So the real R&D focus areas that we have within the group, of course, they're on small organisms. As I've already mentioned, the smaller the organism, the more challenging it is to have a sufficient yield of nucleic acids for all of our platforms from the same individual. So we really need to work to maximize the yield, have strong protocols, and in some cases, we might need to use many individuals in assembly and curation. We also need to deal with the recalcitrant organisms, so those that are particularly unyielding in terms of nucleic acid, whether that's because of the presence of contaminants or inhibitors. But we will need to have new and evolving disruption techniques and protocols in order to be able to deal with the broad range of taxa on our list. And also we are looking to make strong improvements to the routine throughput. We would like to be able to scale the methods that we have in-house already, really streamline those processes and apply best practice from diverse sources and all of our collaborators. Okay. So that brings me to the end, and I've included a photograph of the team with our current COVID-safe working practices. I thank you very much for your attention and welcome any questions.
Thank you, Caroline. And you certainly get the challenging job of working with all that diversity of samples. And I think the question that we've got through really captures that, which is, do you have to adapt DNA extraction according to species, or is it one size fits all?
Thank you. We are aiming to have a very standardized DNA extraction protocol that we would like to apply to as many species as we can. However, there will be a large number that do require more specialized procedures. And that's one of the big R&D projects that we're looking to do, is to work with all of our collaborators in the taxonomic working groups, try to understand where that picture changes, what will be the most challenging groups of species or the most challenging categories, and how we can really develop and refine our processes to make sure that we can sample those in exactly the same depth as we would the more straightforward samples.
Great, thank you very much. And I'm afraid we have to stop there. And I'd like to thank all of the speakers in the session. It's been a very stimulating discussion. And we will resume in just over 5 minutes, where Peter Hollander will be taking over, where we'll be moving to genome assembly and sequencing and data analysis. So thank you very much.